So Mark, as we get started, why don't you tell us a little bit about how you got started in this field and the kind of work that you do on a regular basis?
Yeah, so it was serendipitous, and God definitely opens up doors. We have our plans, and then He directs our paths for sure. So actually, even going to Houghton was a big change for me. I was headed to a completely different direction and went to a Christian retreat, and Lord convinced me that I needed to change directions, and actually applied after the acceptance requirements in May. And fortunately, because Houghton had a rolling admission, they accepted me. And before that, I was headed either University of Pennsylvania or Naval Academy or University of Kentucky. So that was just God last minute opening doors. Well, I'd applied to veterinary schools, medical schools, got into a couple, couldn't afford really, but I really had in my heart doing cancer research. And so of all things, I had done for one of my last requirements, phys ed requirements, I did horseback riding up at the farm, and fell in love with Morgan horses and purchased it for myself for a graduation present, a yearling Morgan stallion. Went home, went back and picked him up. It was up in Western New York, brought him home. And when I walked in, the one place I was most interested in going was Roswell Park Cancer Institute in Buffalo. And I walked in and my mom said, "Look what the post office just delivered. It was lost. It's your acceptance package to Roswell Park." Had to be there 2 weeks later. And I said, well, I made my decision. I have this horse I got to take care of. I'm just going to get a job. And so I ended up going to Roswell Park. My folks said, "You'll get up there," and loved it and really did enjoy. I did tumor immunology, which was really an unpopular area. I used to say it's one area that would accept me. So because tumor immunology was if we think back, Dr. Cooley, who did really the first immunological, he took breast cancer and injected into patients to create antigens. So it was the first immune reaction and actually destroyed some of the breast cancer that way. That was back in 1800s that was done. So that really, he's considered the father of modern tumor immunology. So after that, after graduate school, my mom fortunately sent 100 resumes out, and I interviewed at ICI Pharmaceuticals. Didn't know really what the job was. It was clinical research. I thought I did a great job buffaloing them, and turns out your first job is usually because people like you. They were interested in my prostate cancer research because I was doing learning techniques of prostate cancer, but my real research was ovarian and pancreatic cancer. And funding had ran out, so I left with my master's degree. But interestingly, I had done enough prostate cancer work that they hired me to head up the managerial part of clinical research for phase 2 and phase 3 for a prostate cancer drug. And so able to do that. And once we finished the studies, we had to write them up and do the regulatory filings to the health authorities, both Food and Drug Administration in the US and then also globally. And the God working again, unfortunately, someone developed their second bout of cancer, had to take a leave of absence, and they were in regulatory affairs. And I got to do some of this work, including writing the US package insert for Zoladex, which is a prostate cancer drug that I first worked on, and became very interested in that. The opportunities weren't there at that company. And so eventually it was something that drove me, that I actually left for a small biotech company that was owned by Kodak, called Eastman Pharmaceuticals. And they had moved everybody from Rochester, New York, 100 employees, well, I was employee number 99, to Pennsylvania. And I joined them as part of regulatory affairs. And I learned the regulatory aspects through that position. Well, long, different route. And I think that's the biggest thing, is that God will open and close doors and follow the passion he puts on your heart. And that's really kind of my best advice to any students, is that each one of us, I believe, are created for a unique purpose and accomplishments. And we just have to be open to his guiding to be able to achieve that for his glory. So.
Yeah, interesting how a number of different circumstances in those first couple of jobs led you to get some expertise in some different fields. And yeah, the path just kind of opened up then. So now as you lead teams to attempt to get these drugs to market, what does that look like over the course of a journey like that?
Yeah, so I get started involved at the typically when a drug is we call it a drug candidate. So the early preclinical discovery work is done, and we have a target for a product. Sometimes there is a product, sometimes just a concept and a target. And I start to provide some of the guidance on development. So I really consider myself a drug developer. I'm not creative enough to do discovery. That really takes the art of discovery is looking at things differently. So you have to look at things, the same information from a different perspective, and that's how breakthroughs occur. But the key is that drug development, you can either study a drug and take forever, or you can develop the drug. And so that's my expertise is drug development and knowing what all the steps are. So I take that and start to outline even what is our clinical opportunities, what treatment. So we can say, this is a drug to treat cancer, but there's so many different cancers. And we might look at if it's a targeted therapy with a specific gene mutation, there may be numerous tumors that we could go after. I can look and see from a regulatory perspective, where is the greatest opportunity to have speed to patients as well as speed to approval. Now, the downside is sometimes that's not enough of a return on investment because there's not enough patients. And so it's a balance between here's the opportunities versus here's the return on investment potential. Because unfortunately, once you have your drug approved, your patent is running out. And so if you have a very small niche indication with 1,000 patients total worldwide, you either got to charge a whole lot, which nobody likes, or your patent exhausts until you can get one of the larger tumors such as breast cancer or lung cancer or something where you can charge less and still make a significant profit. Because well, fortunately or unfortunately, it's a business, so there has to be some type of return on investment. And it used to be when I started, only 1 out of 10 cancer drugs ever got to market. We've improved that, and we're still 1 out of 7. So what people are paying for is not just that drug; they're paying for the 6 failures. That and depending, so the sooner you can kill a drug in development versus just studying it, the less costly it will be, and then that all gets accounted for as to what we're charging for a drug that does get approved.
So are you doing scheduling as well in terms of clinical trials and accounting for how long those take and patients' reactions and all those kinds of things?
I help design the clinical studies from a regulatory perspective. I've had the good fortune of being mentored by some excellent physicians in the past, and also other drug developers. It was the one kind of value that I had was always looking younger than I was. So I got a lot of physicians that were towards the end of their career that took me under their wing and mentored me, which was invaluable. I think that's one thing I learned is that I can learn something from anybody, even when first person that started. They may be in it for a week and have a unique perspective. So I will listen to all people and try to pull that from them because I can learn something from them. And likewise, I try to invest in others to provide them that. While mentoring them on other aspects, I can still learn things that have a different perspective on. So that's always key to keep in mind about when you're mentoring, you're also learning. So it's
yeah. I imagine in your field, there's always new techniques, new discoveries to keep up with as a physician would at the same time, new treatments, all those kind of things. So that's probably also one of those things that's in the background all the time is reading what other people are doing, reading new techniques, reading new discoveries.
This year I had to learn about hepatitis B. So I'm an oncology cancer drug developer, but we had a new CEO that joined our company last February. A conference called JP Morgan out in California every year where you speak with investors. And someone put up a slide on a concept for potential treatment of hepatitis B. And someone said, did you realize that Gilead bought Pharmacet for $12.5 billion based on 40 patients in hepatitis C with what's known as a functional cure? March 1st, I was put on a task force. We discovered a product from concept of product in June. We manufactured it by September. We did the preclinical toxicology work in October, November, and December. I helped with the team and we filed it in January, and we are approved now in 8 out of 9 countries to do first in human studies to look for functional cure with hepatitis B. Hepatitis B, there's still residual circulating DNA fragments, so patients aren't cured, so they can have a relapse. Most patients are stabilized with nucleoside analogs, which affect the DNA of the virus in replication, but they're not cured, so they have to be careful because they can have relapse. Over long term, they can end up cirrhosis of the liver and liver cancer. So we were able to get that in 1 year, less than 1 year, first in human, from concept to first in human. But I had to learn a whole nother therapeutic area that I had no clue about. So lots of journal reading and
Yeah.
time doing that. So.
That seems like a lot of checkpoints on the way from having a drug that's conceptually done. So you're the person that's kind of driving that process and making sure that this group is making their deadline and this group is making their deadline.
I have a project management team that reports to me, and they will do the scheduling and the tracking and durations. Also help me with the budgeting for projects. So there's a project lead for each project. I happen to be, for our lead product for this company, Immunocore, I happen to be the project leader for that. And then we have other scientists that are leading. We now have 4 oncology agents in the clinic in various stages, and 1 for hepatitis B in the clinic. And we are now working on a product, and I just heard today that we're actually manufacturing it, so it's a real for HIV, for a potential cure for AIDS, HIV. So we'll be taking that in the clinic by 3rd quarter next year. So, which is another area I'm reading up on. It's very similar to the hepatitis B, but it's dwells differently in cells and the residual, so they can have flare up there. You know, with the nucleoside cocktails, HIV patients have been very much able to have a much better quality of life and also survival. Magic Johnson, probably a prime example of someone that's positive technically, but has lived for 20, 30 years with the disease. But now there's starting to get to the point where there's escape. So there is a real need for a potential for cure for HIV. And due to the immune mechanism that we utilize, we have a lot greater affinity and potential to provide that cure. So that's the next new frontier. So.
Yeah, that would be a world-changing drug. I know one of our alums, Deborah Burks from the class of 76, is normally traveling the world and trying to eradicate AIDS in every corner of the globe. And I'm sure she'll be thrilled to know some of the breakthroughs that are being made now. And I know she spent a lot of her life in HIV research too. So that's fantastic. Yeah. And Mark, you mentioned you have a presentation for us to kind of explain, even for the layperson, some of the science that's involved in what you're doing. So I'll let you share your screen and show us what that looks like.
Can you see my screen? All right. So I work now for Immunocore, and our slogan is science that transforms lives, and to transforms lives. And let's see, am I going to advance this now? This is a pictorial of what we do. So our company harnesses the immune system to fight disease by targeting through an off-the-shelf. So there's recent therapeutic areas has been like CAR T, which is a cellular therapy. The downside to CAR T is that people go through leukapheresis to remove their cells. They reprocess their cells genetically and then grow grow them, and then they are re-administered to the patient, but the patient has to go through a chemotherapy regimen to destroy their immune system before they get their new cells back. Unfortunately, you can have patients that die during that conditioning regimen, as they call it, before they even get their cells back, but they're highly effective. What we're doing is we're creating through the HLA, which is the human leukocyte antigen portion, through the major histocompatibility complex, as it's known in immunology, to intracellular proteins. And so what we have on the right is a T cell, and CD3 is an effector, and in the middle in the blue is what we create. So this is mimicking what happens naturally in the immune system, except we add from a monoclonal this little coil at the end, which is called CD3, a protein that attracts T cells. And on the left here, we have a cancer cell in which the HLA complex, which is in red, and the little gold part is the protein that we're targeting. So we target, and we have an envelope we're indented. And so we're very specific just to that target, which enables us to be very specific, having less toxicity of other cells, because we're targeting cancer cells. It attracts the T cells, the white blood cells that are CD4, CD8 positive polyclonal T cells. And then upon connection with enough synapses, we have to have several synapses. We're able to do typically 4 to 5 synapses, and we can have efficacy. And then granulocytes, which are represented in the yellow, are released, and they kill the cancer cell. So that's what we are all about. This is another pictorial. So a lot of companies on the bottom here are cell surface antigens in yellow, and there are antibodies that are created to that. There's antibody drug conjugates, which is adding a chemotherapy drug typically to an antibody that gets released upon connecting with the cell. There are bispecifics, so that what you do there is you have 2 antibodies that are linked together that attracts cells. And then you have CAR T cells, which are reprogrammed cells, immune cells of the patient that are reprogrammed and then administered back. The reprogramming is done through a monoclonal antibody. What we're doing is on the top part. So when there's a protein, a foreign protein, it goes to the intercellular target, and there's a proteasome that splits these proteins up into small fragments. And those come out on an HLA, human leukocyte antigen, that ultimately goes to the cell surface, and that's how it gets exposed. But these are intercellular proteins, and even protein fragments, which enables us to be very specific. Cell surface antigens, or proteins, only represents 10% of proteins for cancer or any other disease. Because we're intercellular, we have 100% access to any protein. And then we attract. So this next slide shows naturally these exist. The HLA natural protein is exposed on the cell surface of the tumor cell as foreign. The antigens are there. They're recognized as foreign. We can measure them. We can do immunohistochemistry to stain for them. And T cells will naturally go. So a lot of patients can have a spontaneous remission of their cancer because their immune system will kick in. My theory when I went to graduate school was that people got cancer because it escaped the immune system. And that's what I was working towards. And that's probably one reason why you get cancer, because some patients can fight them. You're exposed to free radicals and environmental things, smoking, et cetera, that can cause DNA damage leading to cancer, but then it ultimately escapes the immune system. So what we're doing is redirecting. And so we have this protein in the middle that we create, and we do in between, we do a creative disulfide bond, which allows it to be soluble so we can do off the shelf protein. So it's a fusion protein, and we fuse on an effector, which in this case is anti-CD3, which attracts T cells. We can also put on PD-L1 or PD-1 or any other effector, because in fact, one of the things we're going into next after infectious disease is autoimmune diseases, and we're targeting type 1 diabetes. as a B cell mediated thing. So we can catch children that have the potential of developing insulin dependent diabetes. We may be able to prevent them from ever having diabetes in the first place. So that's our next thing. We need some more funding though to get there, so which is always fun. We do everything through this. In fact, down here is Kintrack, which is my product. We've already manufactured our commercial supply, so I won't belabor that. This is the 5 compounds I talked about. We have partnerships, one with Genentech, one with Roche Genentech, and we also have one with GlaxoSmithKline. And the human HIV immunodeficiency virus, we're actually partnered with the Bill and Melinda Gates Foundation, which has been an interesting experience as well. I'm working on uveal melanoma, which is a #1 malignancy of the eye. It's primarily a choroid portion of the eye. It has very distinctly different characteristics from melanoma. Melanoma is of the skin. Genetic differences, mutations are very different, and there are no treatments. So typically what happens is patients have their eye removed ultimately, and even friends of mine, I see Mark Ura is on there. You know, I asked him this winter how many cases he had seen, and he said, well, he's seen a couple, but they're usually referred to a special area for treatment. Most patients are cured with their initial surgery. However, in some patients, there's high risk due to the genetic mutations, and they will progress to metastatic, and there are no known treatments. So we're working on that. The reason for this is that it expresses an antigen called GP100, and GP100 is what I've targeted. It's also expressed on normal melanocytes in your skin. It's what gives you the pigmentation of your skin, whether you're dark skin or light skin or olive skin. It's related to that, and I'll go over that in a minute. But uveal melanoma, it peaks incidence at 55 years old. It typically, risk factors are light-colored irises, Caucasians, and then there's a PAP1 mutation, which is part of the BRCA1 associated protein, which is also associated with breast cancer. There's only 1,600 to 2,000 patients in the US, and there's about 8,000 patients globally. So it's a small indication relative to the things. Survival, once you get metastatic, there is poor. 50% of patients will ultimately develop metastatic disease. 1 year survival is only 10 to 40%, and most patients will die of liver failure because the target for metastatic disease is through the lymphatics and to the liver. So this is our drug. So these are normal melanocytes, and you can see that the GP100 is expressed, and that's what's in the brown. That's a immunohistochemical staining of the melanocytes showing GP100 expression. In cutaneous melanoma, it also expresses that, and that brown is, again, this is for cancer of the skin, melanoma of the skin. And then likewise in uveal melanoma, it's highly expressed. So what we have done is we have created this TCR redirecting. And what you see here is these small cells are T cells. The red cells that are stained are GP100 positive melanoma cells. And the GP100 negative are in blue. They're a little larger, they're bigger than the T cells. When you see a burst of green, that cell is dead. So apoptosis has taken place, and the T cells have killed. So this, if anybody remembers Pac-Man, this is my version of Pac-Man. And you'll start to see green light ups. That cell just died. That cell just died. It's in yellow. That cell and that cell just died. They were killed by the T cells. So that's our development. From that, this is a patient at baseline. They had no T cells in their tumor. The green and magenta are T cells. After treatment, at cycle 1, day 2, 1 day after treatment, a lot more red. And at day 16 after well, just be their third treatment because we treat weekly. Second treatment rather, a lot more red. And likewise in this patient at baseline and then day 16, the hard part here is you have to have patients that are willing to get more than one biopsy, and this isn't easy to get. With melanoma, it's a little easier than it is with uveal melanoma because it's in the liver, so you have to be willing to have a liver biopsy. I've got 2 studies I've designed. One was a single arm study to look at patients that were refractory to all therapies, in which we could have a strategy based on response rate for approval along with duration. And then a randomized study in which we randomized patients 2 to 1. For every 3 patients, 2 patients were randomized to our test drug, and 1 patient was randomized to investigator's choice of, unfortunately, drugs that are utilized, but they don't work very well. So I have the results for this study, which were promising. Due to time, I won't take a lot of time to present that. We'll see how much time we do have. And I am waiting anxiously. We've accrued all the patients, 367 patients to this randomized study. And I'm waiting for, unfortunately, it's an event driven. So we have to have our first analysis is after 150 deaths. So it's sometimes morbid, and I always remind people that it's individual patients' lives that we're dealing with. That will trigger the first analysis. 200 patients will trigger the second analysis, and the final is based on 250 deaths. And these are heroes in my mind because they are willing to potentially have no benefit to them as individuals, but are contributing to science. And still today, in cancer, there's only 4 to 5% of cancer patients will ever participate in a clinical research study. But those patients really do benefit. One of the things I did work at Bristol Myers, and Opdivo is advertised on TV quite often. And one of the things we did, because I reviewed all the ads and the commercial part to make sure it's consistent with the labeling, we did thank the patients and their families for participating in the clinical studies. And that is the one thing, and that's what drives me more than anything, is the patients. One more day that I commit to doing work on this is potentially one less day that a patient has to suffer and his family members. And with Opdivo, I know we have a lot of cures for a lot of cancer. And I mean, these are patients. I know a woman on Yervoy, first immuno-oncology I worked on, met her. She was diagnosed 4 months before her wedding with stage 4 cutaneous melanoma, given less than 6 months to live, and she was 4 months from her wedding. They decided to get married anyway. She went on Yervoy and became cured. She was overweight then. She became a marathon runner. She now has 2 children. One is 10 years old and one is 6 years old. And she is cured, disease-free, when she was given less than 6 months to live. And that has been one of the most rewarding things, was meeting her and getting to talk to her about her experience. And really what drives everything I do is the patient. In fact, everybody that works with me and the teams I lead, they know that that's what I bring up every single day. That's what motivated me to go into cancer research. And it's what motivates me every day, is to keep persevering and contributing that way. So I'll end it there, Dan, because I know you may have other questions, but.
Well, I'll say that you know your stuff, Mark. I mean, I give you an A for the presentation. And the other thought that I had is I just really should have paid a lot more attention in biology class.
I'm sorry, yeah, I lost you with a lot of.
No, I am glad that you're doing this work, and it's neat to hear that. Well, one of the things that did strike me was that these are very targeted, specific tools that are very effective if they're used the right way.
That's right.
And I imagine that part of your work is working with physicians as they diagnose, making sure the diagnosis fits the drug properly. How do you help a doctor make that match? Or is that more on the pharmaceutical sales rep?
No,
or
some of it's through drug development. So we do have biomarkers. So there's genetic abnormalities. Actually, now through there's a company called Foundation of Medicine that I work with. They can run. It's rather interesting. It takes 3 days to run your DNA completely for your body on their system, and then it takes 15 days on a supercomputer to crunch it to know what all your genetic abnormalities are for an individual patient. Patients are now tested that way. So we call it targeted therapy. There's tyrosine kinase inhibitors that are very targeted. Pfizer has one for lung cancer called crizotinib. It literally represents about 2% of lung cancer patients, but if you have that abnormality, you're pretty much 100% guaranteed of a long life by going on this oral therapy that you'll take for the rest of your life. But it's what's we call them driver mutations. They drive the cancer. In my case, I'm looking at GP100, which is an antigen, so we can stain biopsies to see whether the patient has prevalent enough. Likewise with Opdivo, the interesting thing there was it's a PD-1 inhibitor. Scientists do have a sense of humor. PD-1 stands for Program Death 1. So it sounds a little like Star Wars.
Yeah.
So actually a Scottish man that still works in the lab with his kilt on discovered that, and actually Yervoy is ipilimumab, which was a checkpoint inhibitor called CTLA-4. Jim Allison 2 years ago won the Nobel Prize for that, and he's a very interesting individual from Texas, University of Texas, San Antonio is the labs he's worked out of, and has a passion for cancer as well. His mother died of melanoma. He made a personal commitment as a child to do that, and it's actually very similar story to mine. It was my aunt actually, which actually led me to Houghton as well. My aunt, when she was 32, died of lung cancer when I was 16, and my father was actually transferred. And she was like my second mother. She was not married, and growing up as a pastor's kid, we didn't have much money, but she was able to give us some nice things, clothes, and take us out and do things. So when she died, I was very angry, and I was angry at God and really wanted to do something about cancer, and that just haunted me. And God changed my heart from an anger to a passion and a compassion for people with cancer and a drive. And I think that's just part of his plan for my life. And that's why I had, nothing fancy due to my own of being successful, but it really was, I believe that was my purpose, was to get into cancer drug development and use my gifts not only in science, but also motivating people and my skills, which I've developed over the years. And you never know what class is going to be most important to you. And I think I shared with you one time we got together. It was small group dynamics with Professor Ruth Hutton that constantly comes to mind about the dynamics of a small group, which is really what a team is. And you get the forming, performing, storming, reforming, and outperforming. And I will actually throw in artificial problems into a team so that they have to actually storm and then reform, and then they start outperforming what they do. So we actually create a lot of that as senior leaders, challenges, so that they actually will have to come together because they end up, sometimes it's the senior leaders that are their mutual enemy, so they bond together against us to defeat us in the cancer. So whatever, but that's part. And so you never know what class, I took it as to finish up my, I think it was my junior or senior year, to finish up my requirements as a liberal arts student. And I'm a firm believer of liberal arts because that is what enabled me to be able to have also the business. I worked some brilliant MD PhDs. Sometimes I have to wonder how they get to work in the morning, but brilliant. So I compensate for their brilliance by giving practical. And the other thing I always recommend to students is number one thing that will drive your career is your communication skills and ability to communicate with others, both verbally and written, and also to be able to lead teams and work within a team environment. That those become key in success.
Yeah, one of the questions we got, Mark, is kind of along those lines. One of our listeners asked if you were to create an interdisciplinary major at Houghton, what would that look like? Based on the work that you've done in your life, that kind of intersects science with team leadership and management and project management and everything else. What would that look like?
Yeah, I definitely think leadership is something that is, some people have it natural, but there is an ability to really develop that. And I think if I was to do an interdisciplinary leadership, there's a few schools that actually have leadership majors now, which is actually something that is very much needed in all industries and in general. I think the biggest thing that the world need is leaders with integrity. A lot of companies 10 years ago did not have integrity as one of their main values. I would say every company has integrity as their number one value for leaders, or one of their top things. Where they struggle is actually following that. There's a lot of things, and I've left a couple companies because of lack of integrity of the senior leaders, which ultimately could have reflected on me. So I ended up leaving those companies and moving on to another company because of that. It was all about the patients. For them, when the going got tough, it was all about their bonus. And it really hadn't, and they were hiding a lot of stuff. I mean, to the point where we were doing genetic testing, and this is how I got to know Foundation for Medicine. It was labeled that they were a female, and the genetic testing came back male, and there was a label error, and they tried to compensate for it, which was impossible. So they had to. I got them to start over again, get another sample, and make sure it was labeled properly. But that was the type of thing like, oh, we'll just relabel it. Well, you can't. You don't know whether it's an accurate sample. So that integrity in science has to be key so that you can trust those results. So.
Yeah, Mark, I'll ask one of these last questions. What advice would you have for one of our students who's interested in a career in the pharmaceutical industry? What should they be doing while they're at Houghton, and what should they be planning on doing?
Yeah, so a liberal arts education is excellent. I mean, I think that's one of the keys. And there's multiple opportunities. It doesn't have to be just science. My daughter, Erin, who's I think listening, she's involved in, she was a journalism and communication double major at the University of Richmond. And she took enough science that she could understand it. And now is doing press releases for pharmaceutical companies, promotional material, helping to justify pricing based on value added, things for like genetic therapies for hemophilia cures. What does it look like if you're charging $2 million for a product, but yet the person's cured of a very serious debilitating disease? So she's involved in that. And so there's a lot of opportunities in pharmaceuticals, regardless of whether you're science-based or not, every aspect, finance, et cetera. But the key, I think, really is networking, and then taking opportunities for internships. I think that's another thing. There are a lot of internships. And then don't be discouraged. One of the interesting things about my company is we'll pay for, if you want to go to Oxford University, we'll pay you for your career, and you can go at night to Oxford University to get your PhD. And they'll give you time in the labs to do your research right on site, so you don't have to even go, and give you time off. So you work for us 3 days a week, and you do your lab work for 2 days a week, and in 5 years you have your PhD, and it's been paid for, and you were getting paid a salary all the way through. So there are other innovative ways that you can actually finance your future education. So don't be discouraged if it looks like it's astronomically impossible financially or otherwise. Most companies will have continuing education and will support that.
Well, Mark, I appreciate it's not every day we get an award-winning pharmaceutical researcher and leader to talk to us, and we just appreciate what you're doing. I'm incredibly encouraged to know that you're out there doing the work that you're doing as well. I know these diseases and illnesses, they affect our family members, our loved ones, and none of us know what our futures look like, and if we might be benefiting from one of these therapies, down the road as well. And I know with a pandemic happening right now, I know your company's working on some things in the background there too, in terms of vaccine development and treatments for later on. So we just grateful for you being here tonight. And those of you who are listening, I encourage you to tune in again on November 11th and hear from Denise Jones. But thank you for being with us tonight. And thanks again, Mark, for taking some time out of your evening and doing another Zoom call with us tonight.
That's right.
And I look forward to when we can get together again.
Thanks, Dan. Thank you
Yep.
for inviting me, and it was a real pleasure. And if any students want to contact me, I'll give you you have most of my contact information. Do that. I'm happy to talk with them one-on-one, and to see what doors might be able to be opened up to them, especially if they're interested in a career in pharmaceuticals. Nice part about 34 years in the industry is I've got a network out there that's huge, as people have gone from different companies. It's probably a pharmaceutical company that I don't know somebody at this point in time. So that's a key to getting your foot in the door often. So use it. That's what alumni networking is about. So I'm very open, and I really enjoy that aspect. So
All right. Thanks, Mark.
thank you.
God
Appreciate it.
bless. Thank you, everyone.